お待たせいたしました。お時間になりましたので、これから最後のトークプログラムを始めさせていただきます。本プログラムは英語で進行させていただきます。こちらで視聴いただく方には、同時通訳の機器を配布させていただいております。チャンネルを1に合わせますと、日本語訳が聞けますのでご利用ください。もうすでに設定がされていると思います。オンラインでご視聴の方は AI 通訳による字幕を表示しております。実験的な取り組みとなりますので、動作に不具合がある場合がございます。ご容赦ください。So, let us begin the talk session. Good afternoon, everyone. As it is time, we would like to begin the final talk program of the day. First of all, thank you very much for joining us today. In this session, we are welcoming guests who have contributed to the two project exhibit in the next room. Indivisibles, the hidden life of microbes, and the garden experiment. This talk will be co-hosted by myself, Tomoko Sekiguchi. I'm festival advisor for Matsudo International Science Art Festival and member of MISAF Lab. And together with Anne-Mai Do from the Science Arts Technology Office Tokyo, Embassy of Switzerland in Japan. Anne-Mai, could you please introduce yourself and tell us about the special guest joining us from Switzerland?
Thank you, Tomoko. So as she just said, my name is Anne-Mai, and today I'm very happy and honored to represent the Embassy of Switzerland in Japan, and more specifically, the Science and Technology Office Tokyo. And our role, very briefly, in this project was to bring the amazing partners together and then help with the coordination of the efforts to what led to bring us today together. And so joining us from early morning Switzerland, we have a very special guest today with us, the NCCR director, Professor Jan Roelof van der Meer. And Jan, could you please introduce yourself very briefly?
Yes, sure. Thank you very much. Welcome. My name is Jan van der Meer. I'm joining from Switzerland on the other side of the planet, where it is still morning and rainy, but I'm very happy that we can have this joint talk and also this joint work on the Soil Citizen Garden project that we will speak more about. Thank you very much, Anne-Mai. Thank you very much, Yoko. Thank you very much.
Yes, thank you very much for the introduction. I am also joined by my colleague Yoko Shimizu, co-founder of this festival and bio artist of MISAF Lab.
Yes, thank you very much for the introduction. Thank you everybody for joining us in this art science talk. And I am very honored and very excited to be able to launch a special joint research project with you this year. And I'm looking forward to the panel discussion today.
Thank you. Next, Yoko, could you please give us an overview of the exhibition and the project?
Yes. So this year we started a collaboration with the NCCR microbiome team as well as the science technology team from the Embassy of Switzerland. This year there are mainly 3 collaborations. The first one is the traveling and satellite exhibition of the Invisibles, the hidden life of microbes. And then the second element, which we are very excited to launch this year, is a citizen science art project called the Garden Experiment. This year, we also have the Science Art Talk event, which we are currently hosting right now. So for those of you who have not seen the exhibition yet, currently on the other side of this main hall, there are 3 exhibition rooms. We have connected all 3 rooms to tell the entire story of the microorganisms, what they are, why they are important to us as humans, as well as for the entire planet. So in this traveling exhibition, you will be able to learn about the basic information of microorganisms. And as you go through the exhibition, in the end, you will be able to see the launch of our joint research project called the Garden Experiment. So in the garden experiment, citizen scientists in both Switzerland and Japan are participating to conduct a large-scale citizen-based soil microbiome experiment together. This will be a 2-year collaboration where the citizens actually already transplanted the soil in Switzerland and in Japan in their own gardens and fields. And in November, we will actually collect those samples, analyze them, and next year in MISAF 2026, we hope to showcase the result of the experiment as a large artistic installation. And we are very excited to be able to today discuss a little bit more about the Citizen Science Art Project.
Thank you.
Thank you. Now I invite Jan. I think you have a small presentation for you. If you could share your screen, and I give you the lead.
Thank you. Yes, let me share this small presentation with you.
Jan, can you hear? Yes, perfect.
Can you see the presentation?
Yes, we can
Yes.
see it. Thank you.
Thank you. Yes. So first of all, I would like to welcome you to this presentation, and also for the honor that our Swiss National Center of Research in Microbiomes is invited for this collaboration with Japan. I think this is a great honor. My name is Jan van der Meer. I'm the current director of this NCCR. Here you see a picture. We are about 150 researchers in Switzerland across different institutions who is researching the question of what are microbiomes. And as a microbiologist, I work with bacteria. I will show you that in a minute. But our realization is that all our life, as we know it, is not just a human being or a cow or a plant or the soil, but everything is covered with microorganisms. Whether we like this or not, we have to live together with the microbes. And it's more than that. They play an essential role for us. They play an essential role for our health. They also make us ill from time to time. They also provide nutrients to plant growth. They recycle all the material on our planet. And this is the major question that we try to understand. How is this life connected? How is this life connected with us? How is this life connected, and how is this life formed? I don't know if you ever saw what are microorganisms. Here on the left part, you see cells, small, black, like shaped, spherical, and long objects. These are microbial cells. And you can see that they move. You can see that they divide when we give them something to eat. When we give them something to eat, they can grow. They can form structures that we call colonies. That is a group of the same microbe together. We can study them in the lab. You can also work with them in the environment. For example, on the right, you see a picture of microorganisms that live in a wastewater treatment plant. We use wastewater treatment to get rid of our organic waste before it enters an aquatic system. And essentially, we feed bacteria and we feed microorganisms. There's a huge diversity of different microorganisms. It's impossible to determine them all. We can take a scoop of soil and analyze what are the microbes that are in there and represent this as sort of a bar diagram. So all these small vertical lines that you see in green and in blue represent different species that are present in just a scoop of soil. Thousands and thousands and thousands of different species. And we have no idea what they do. We have no idea why they are there. We have no idea how they live together. So the aim of our project in Switzerland is to understand how all these microbiomes form, how they develop, how they function, how they live together, and why that is important for us. We also aim to understand and develop methods that may help us to restore this diversity and this equilibrium between the microorganisms and us. When this is disturbed, for example, when you are ill, but for a soil or a water, when this is ill because we have polluting substances, chemicals, or when they are diseased, or when it is too dry, or when it becomes too warm, or when we have bad practices by industry or agriculture. Then the microbial diversity is disturbed, it declines, and we need to be able to restore it to a healthy environment again. This is where the Soil Garden project comes in, because the project is an example of a method that uses microbial transplants. Transplanting healthy communities from one side may help to restore others and bring new diversity. So soil is like a home for bacteria. Soil to us looks brown or dark or black, but in fact it's a collection of all kinds of small particles filled with water or air, filled with roots, with earthworms, with fungi, with nematodes. And if we go down in scale, we see eventually the microbes in their small pockets, cells living together, eating from the soil particles, eating from the roots, eating from the old material. Now, a transplant would mean that we take a bit of this soil, the home of the microbes, and put it in another home. And by mixing this, the microbes that come from one place have the chance to migrate to the others and will slowly grow into their new home. Now, why do we need a soil cohort? We could do that in the lab. And why do we need citizen science? The problem is if you want to test a transplant, a new home from microbes of one to the other, there's a lot of variation that may appear. And this you cannot control in the lab. By testing many, many different new soil homes that citizens provide us in their garden, we hope to discover what the transplanted microbes like to live on and why they develop. We can also study which of them migrate and survive best by doing the same method that I showed you before with all these vertical lines and all these species, and which factors actually control what the resulting new community will look like. Are we successful in making new diversity? Are we successful in making a new healthy soil ecosystem? These are questions that we want to test in the Citizen Science Project and the cohort where we have many different people and many different soils that we are testing. The Soil Garden Citizen Project is also, for me, embracing a friendship among people, because we like science and we like to explain to people how we can reach out with science and combine things that are both artistic, have a function and have a purpose for our planet, and also art. So I'm very happy that we can connect the National Center of Competence in Microbiome Research in Switzerland with the Matsudo International Science Art Festival in Japan, and also my dear colleague Ryo Miyazaki, who on the Japanese side will help with the analysis. How are the two citizen projects different? In the Japanese part, the transplant community is a natural living compost. At the Swiss part, our transplant is a regrown natural soil community that we have grown in large containers before, and we have followed how this soil community is developing, what its diversity is, and now we are testing how this can survive in different homes. With that, I would like to thank you a lot. And I think that this project is also about caring about our planet, because caring about our planet starts with life in the things that are nearby to us, such as life in the soil. It's not just a material that we use. No, it's a living matter. Thank you very much for participating in the Soil Garden Citizen Project, and thank you very much for listening to my presentation.
Thank you very much, Jan, for your inspiring presentation. And now I would like to start the discussion, and maybe with a question to you. So now that the project is progressing, could you share maybe with us what kind of scientific and social outcome are you anticipating, and how you just mentioned there is a bit of a difference between the experiment in Switzerland and in Japan. So how do you plan to compare these microbial data sets so between the two countries, and do you expect significant differences between the two countries? Yes, thank you.
Yes, thank you very much, Anne-Mai. Yeah, these are very interesting questions and also very difficult for us. First of all, there is a difficulty to the project because one part is in Japan and one part is in Switzerland. And when I discussed this with Rio, my colleague, we first thought, oh, we can transport soil from Switzerland to Japan, but you cannot. And you cannot transport soil from Japan to Switzerland. You cannot. So that is when we decided it's better to work with material that is present in both countries. The other thing that we need is living material. So we need to have a living material that makes sense for people to work with. Because when we talk to citizens, they don't want to be just, let's say, doing an experiment for us. They want to understand, why do we do this experiment? Why is that good for them? So people know very well what is compost and why you would add compost. But strangely enough, what is not very well understood is what happens when you add compost. What happens to all the bacteria that are in compost that you can add? So with Rio, when we discussed, we thought, oh, the best way that we can launch this project is to have two different soils. One with the compost that has the advantage that people know what is compost, why would that be good for a soil? And this is also a very well accepted method by, let's say, farmers, by other, people that grow vegetables in their garden to enrich the soil and hope that it becomes better in its quality. So that's why that acceptance is very good. On the Swiss side, we had the advantage that we also wanted to understand what are the bacteria that we are bringing in to the soil. And when we take a compost, we don't know which bacteria we are bringing into the soil. But because here in our Swiss research network, we were actually growing soils ourselves, we understand very well what that soil is and which bacteria are there. And that makes it easier for us to actually follow which bacteria and which microbes go from their starting home to the new home. Because as you can imagine, what we do in the end is making this barcode of the soils with all these thousands of different species. But then we have to be very careful in saying this comes from here and this originated from there. And that's a very complex scientific problem. And that is why we need to have a very good control on the starting material in one hand and the starting material in the other hand. We also hope that by having two different parts, one in Switzerland and one in Japan, that our study together actually gains in strength because we can ask two different questions at the same time. And for the rest, we are extremely happy that, so in Switzerland, we have more than 220 participants currently. And in the Japanese side, I understood you have 120 or so participants, something like that. So that is sufficiently big to actually do a good comparison of what we want to achieve.
Thank you, Jan. And so speaking about citizen scientists, I would like now to turn to Tomoko, because you have put a lot of efforts into gathering all the citizen scientists here in Japan. And I'm very happy, by the way, to see one of our citizen scientists in the audience. Thank you very much. And so my question to you is, how was the invitation received by the citizens here? What was their response? And how were the local communities involved in shaping this project further?
Thank you for asking. I was truly surprised by how many people were eager to take part in this project. A diverse group of participants have joined this experiment, including farmers, elementary and junior high school, communities involved in forest conservation and riverside activities, and cultural institutions such as the Tojo Day House. We also received wonderful support from the city of Matsudo in carrying out this project. Their understanding and openness towards such forward-looking projects are very important not only for this project, but for the entire festival. When we regard government, businesses, and all residents as citizens, this project has provided a wonderful opportunity for everyone to think together about global challenges and to make contributions to scientific research.
Thank you very much, Tomoko. And now looking more into the future of the project and the next year's phase, as Yoko presented before, the findings or the outcome of this project will be then translated into an art project. And Yoko, you have already explored beautifully the world of microorganisms through other works you did. And so I think we are all very curious to know, how do you blend biology and or science and art, and what are the challenges you encounter when working with something that is invisible and so complex?
Yes, so I think in the garden project, I am very excited. I was working together with Tomoko in launching the Citizen Science Project in Japan, designing the kit together with Yo. I think the design element was also very interesting for us, being able to take a very interesting and very scientific research protocol and turning that into citizen-friendly kit manual, creating tutorial videos. And I was also very, very similar to Tomoko, very, excited and surprised and happy to see so many people participating and working with us. We actually had a kickoff event where we were able to meet the citizen scientists face to face and start the first step of the experiment, which was labeling the samples together. And I think for me, I am also very curious about the social cultural part of the project, where each citizen, each garden, each farm or field is very different. The plants that they're growing, the garden that people are curating with different flowers, visually different, but also different crops that are planted. And also in the Tojo Historical Park, this is also a cultural heritage. So the people who are conducting the experiment, the gardens that are being used in the experiment, that cultural, artistic part is also already very, very interesting. So together with the scientific data that we receive, I am very excited to be able to create maybe next year a large-scale, perhaps citywide exhibition of all the gardens and the people and the culture, as well as all the microbes that we find throughout the city and in the surrounding area. I've always loved nature since I was little. When I was very, very small, I was born in Kyoto, so I was surrounded by traditional architecture, art, as well as bamboo forests and rice fields. And to me, there was always a natural blend of nature and science, exploring nature, and then art. So for me, combining those two fields, biology and art and design, was very natural progression for me. So now in the biodesign or bioart practice, I do work with microorganisms to create different materials, or work with plants and insects to interact or work with bioluminescence to find new forms of communication or expression. I think to the question, what is the most difficult part or challenges? I think the difficult part is, of course, when you work with something that is alive, you cannot turn it on or off. You cannot control them 100%. But that's exactly why I think it's so beautiful and so wonderful to work with. I actually see them as co-creators. I think throughout the history of biotechnology, from ancient sake to modern medicine, I think humans have always co-created or collaborated with microorganisms. And I think the fact that you cannot control something, but it is such a beautiful co-creator with a superpower that you don't have, because all microorganisms or living things have very unique powers and capabilities that humans do not have. So I'm always amazed by the result and the outcome, because I also although I designed or started the project, I also don't know the outcome. And I learned so much, and it's so inspiring as well as beautiful.
Thank you. I have one question for you, Mai. From your perspective, what were the key moments in building the Swiss-Japanese partnership behind this project?
Thank you, Tomoko. That's actually a very difficult question, because when thinking about it, there were many key moments, and I think I would need like 10 more minutes to talk about it, which we don't have. So like from starting from the first time we visited the museum in Switzerland, so the very nice exhibition there, that sparked the inspiration for this project for us in the first place. And then I think also the first meeting we had with Jan's team, the NCCR and you, when Jan mentioned this garden experiment they were about to launch in Switzerland, and then it became this collaborative product with Japan also. And I think more than key moments, I really want to take maybe the time to also express appreciation to all the key partners. So here we have, of course, the NCCR Microbiome, MISAF Lab, but also the ones that could not join us today. Jan mentioned Rio Miyazaki before. He was also instrumental in bringing in sponsor for the DNA extraction phase, which is very costly also. So the Seibutsu Giken that very kindly sponsored this part for the Japan part experiment. And then, of course, the compost companies, BTEC and Musashi Kotahi. And we also got a grant from the Sakai Stunsi that believed in this project. And finally, you mentioned the Matsudo city. And so without all these key players, this would not have been possible. So yes, this brought us today and hopefully next year. And I think we are very much looking forward. And so now speaking about MISAF 2025, sorry, I don't have. I would like to come back to the theme of this year's festival for my last questions to my three fellow panelists. So the theme this year was Delta of Creativity, and I think creativity can also be seen as thinking out of the box. And as humans, we often think in a human-centered way, which is very natural, of course, but this also has led to having a very deep impact on our planet and on our biodiversity. And there is this concept of planetary thinking, which invites us to shift our lens towards a more, not just seeing the Earth as a backdrop of human activities, but as seeing how it's how the Earth is a complex living system. And so my question to you is, how can we shift towards this kind of more planetary thinking? And would you have any advice to nurture this view? I don't know who would like to start. Maybe Jan.
Thank you. Yeah, this is a very important question, no, but also very difficult, because as Yoko was mentioning, microorganisms, they do their own thing, and we can try to understand what they are doing, and we can try to work with them rather than against them. But unfortunately, we have created conditions on our planet which will also change the activities of microbes, because they will find different places to live, they will find different conditions, and that will change how they live things. So for me, planetary thinking in first instance means that we make people aware that there is actually life out there. There's other life than trees and plants, animals, and human beings, which is very important. So already that, I think, is a very big step if people understand, okay, soil is not a building material, soil is alive. A lake is alive. The marine, the sea is alive. We are all alive with microbes as well. So if we already understand that, then maybe we get a step closer to understanding what means like a healthy, equilibrated life for us, for our immediate surrounding, and maybe also for our planet. And then we can hope that the activities of microbes will help us to return to a favorable state for all the life forms.
Thank you, Jan.
What do you think, Anne-Mai?
Oh, I didn't expect that. So I personally don't feel entitled to give any advice, but I think to the start, I would think to have this, enhance this planetary thinking is to stay, first step is to stay curious. And I think already the people joining this festival, they are curious about art, science, and that's a very good start to nurture planetary consciousness, awareness. And I think that's the first step that can lead later to think more as a complex and interconnected world. Yes.
Thank you. Okay. From our perspective, it is important to build daily relationships with the local community that encourage people to participate and to create opportunities that spark interest in meaningful research. We are truly grateful and honored to have the support of so many catalysts.
Yes, for me personally, planetary thinking is using your creativity to think beyond yourself, thinking about not just other human beings, but other non-human beings. There are so many organisms on the planet living with us, but they sometimes communicate or interact in a way that humans cannot perceive. Some of them are invisible or inaudible. So I think it is important for us to find creative ways to sense or understand or interact with them so that we can build a more sustainable future and build empathy towards one another, whether it's another human being or other species. And I think in the garden project, working with so many citizen scientists and also thinking about the soil and the microorganisms living in the soil and the act of actually touching the soil and mixing the soil and connecting with the soil, I think it already builds this connection between you and the planet. And therefore, open research projects like this also helps us not just learn, but to also connect with the planet. And perhaps this type of research, as well as the final outreach, will help us, will help people together from different parts of the world towards a more planetary-centered world. Yes.
Thank you very much, Yoko. I just checking if we have time to take maybe one question or one or two questions from the audience, if anyone has.
あ、そうですか。はい。じゃあ日本語で喋ります。今回の実験に参加させていただいてるんですけれども、ちょっと聞きたいことは、先ほど科学とアートについてということで、確か、何ですか、実験のプロンプトを市民の方に落とし込むというようなお話があったと思うんですけど、ちょっとそこのところが具体的によく分からなかったんですけども、もうちょっと説明していただけないんでしょうか。
Yes, so Jan, yes. Jan and Anne-Mai, we have a question from one of the citizen scientists who has been participating in the project. And we mentioned before about bringing this research and experiment protocol to the citizens, and how we use the design process or different ways of conveying the experimental steps to the citizens. And I think his question is that he wants to learn a little bit more about the process of bringing this research to the citizens. So I think from our side, I know from our side, the process was first Jan and the NCCR microbiome team came up with the first initial idea on how to run this large-scale citizen science project. From our side, as creative professionals, we really wanted to make sure that the scientific research can actually be implemented accurately, but easily. I don't know if easy is the correct word, because I do think that it takes an effort to take part in this experience. But as a non-scientist, for non-scientists, how do you make this experiment more accessible? How do you design a process or a kit or tools that anybody can use in their own garden, so that anybody who is interested in participating can take part in this wonderful scientific research. So I think that was one thing that we tried to be careful of, and Tomoko also did a very wonderful job in communicating that, not just on the website, but making videos and also talking to you face to face during the kickoff events. And I think it was also a lot of a learning experience. I'm wondering how it was for the Switzerland team. Yes.
Yeah, thank you. It was to a large extent the same. So we tried to design the experiment such that it would be robust, because we cannot ask citizens like, take us a sample every day and send it back. That's too complicated. So we tried, at least for now, to find a form where we are relatively certain that the sample we get back is something that has a great value. And we also presented first the people with video and instructions and materials on how to do it, what to do, make it as easy as possible. And we will also, once we have results coming in, we will also convene as many people as we can to the museum here in Switzerland to discuss with them what do these results mean. Because as was mentioned a little bit by Anne-Mai in acknowledging the contribution of sponsors, actually looking at microbes, even in soil, is very, very expensive because we don't look at microbes, but we look at DNA. So if we have 500 samples, then we have to isolate DNA and purify the DNA from 500 samples. And then in that DNA, we are going to look very specifically for signs of microbial life. And then we are going to determine by a process that is called DNA sequencing what the differences of the different microbes are. And this is very costly. And it is also taking a lot of time for even scientists. This is a complicated, very, very complicated step, actually. And that is why it is wonderful when many people participate, because then we can also convey this whole process to the citizens who participate. And I think that makes it double wonderful. So people get an idea what is the life in the soil. They also get an idea what is behind all the steps that you need to understand even such a simple thing. And then by comparing everything, we hope that we get a better idea, how does this life behave in different places?
Thank you, Jan. Do we have, I think maybe...
Yes, thank you very much for your question, and thank you everybody for your participation. I think we're almost at the end of the program. こちらのお時間がもう近づいてまいりましたけれども、本日皆さんこちらのトークセッションに参加いただいてありがとうございます。で、実際に市民科学者としてプロジェクトに参加いただいた皆さんも本当にありがとうございます。私たちとしてもこのような大規模な国際的な学術研究を市民の皆様と一緒に実行することができたのも皆さんのご理解や情熱やあと好奇心、Anne-Maiさんがおっしゃってたように好奇心があってこそだと思いますので、このプロジェクトや実験自体は、また来年まで分析を得て来年の展示まで続きますので、また引き続き皆さんと一緒にこの研究を進めていけたらいいなと思っておりますし、私たち自身もラボとしていろいろな開発も科学と芸術の丘として開発も務めておりますけれども、また今後もますますさまざまな科学と芸術にまつわるプロジェクトですとか体験などを皆さんと一緒に作り上げていきたいと思っておりますので、そのようなプロジェクトがまた今後ありましたらぜひご参加いただければと思います。本日はありがとうございました。